accumulation within the heart, and so on and so forth. And so in the past couple of years, we've had some contributions to this field where we found that the exposure to branched-chain amino acids can influence skeletal muscle insulin sensitivity and can also influence lipid deposition in the heart by influencing substrate utilization in those tissues. And we've also identified that one of the major regulatory nodes for branched-chain amino acid oxidation within the liver is also intertwined with glucose sensing and the lipogenic machinery. And so we're learning that there's more interconnection between these amino acid metabolites and other metabolic pathways than was previously described. So why do I want to tell you about branched-chain keto acids? Well, as I mentioned a slide ago, the branched-chain keto acids, or as we call them, the BCKA, are the first intermediate in branched-chain amino acid metabolism. And they're interesting intermediates in that the branched-chain amino acid metabolic pathway, the first step is a transamination reaction which produces these. This is where you lose the amine group. And then the branched-chain keto acids can stay within the tissue that they were produced for further oxidation. But because a lot of tissues don't express very high levels of this next enzyme, the BCKDH enzyme, which decarboxylates them, this is the first irreversible step and often described as rate-limiting step of the pathway. Branched-chain keto acids often head out into circulation where they head to other organs, mainly the liver and kidney, which have higher levels of BCKDH activity and can further oxidize them. So when we're looking at branched-chain keto acids, we have to think of them as not only an intermediate within a tissue and within a direct pathway, but also intermediates that are shuttled across pathways. And so their levels in circulation are not only reflective of the overall branched-chain amino acid levels, but can also be reflective of perturbations in their catabolism at other sites. And what we know about the liver, which is one of the primary sites of their oxidation systemically, is that in a situation of obesity, non-alcoholic fatty liver disease, diabetes, BCKDH, the enzyme that oxidizes them, gets hyperphosphorylated and turned off. So the liver no longer oxidizes them. And so you kind of lose the normal ratio of the keto acids to their branched-chain amino acids because the keto acids start to accumulate. And they accumulate within tissues, but they also accumulate within circulation. And so this step is controlled by a kinase called BDK, which we have a small molecule that can regulate it called BT2, can inhibit it. And so increases in BDK with obesity within the liver lead to phosphorylation of BCKDH and that gets turned off. The other enzyme that regulates this phosphorylation step is a phosphatase of the protein phosphatase M1 family, PPM1K. And this will dephosphorylate this site, leading to reactivation of BCKDH. And it's in essence, the balance between these two enzymes can control systemic levels of branched-chain keto acids because they control the activity of the enzyme within the major tissues. So when the balance is perturbed and pushed towards BDK being predominant in situations like obesity and insulin resistance, what happens is this gets turned off and the branched-chain keto acids accumulate and they end up accumulating circulation. And so we've been curious about what function they might play in other tissues under these circumstances, such as the heart. And what's known about the heart, at least in healthy animals, is it suggested that it has very high levels of BCKDH protein expression, but it's also the tissue with the highest expression of the kinase. So it's unclear whether or not there is actually very high levels of branched-chain keto acid oxidation within the heart and what an impact exposure to higher levels of circulating branched-chain keto acids due to situations such as obesity, non-alcoholic fatty liver disease, or insulin resistance might provoke within the heart. Our interest in the branched-chain keto acids within cardiac function were further stimulated by this terrific paper from Haipeng Sun and in Yibin Wang's group, where they found that with progression of heart failure, multiple enzymes in the branched-chain amino acid catabolic pathway within heart tissue are downregulated, and this results in an accumulation of our branched-chain keto acids within the heart. And so KIV is the branched-chain keto acid from valine, KIC is the branched-chain keto acid from leucine, and KMV is the branched-chain keto acid from isoleucine. You can see all three accumulate equally. And so they actually went on to perturb branched-chain keto acid metabolism within the heart. And they did this by getting a ppm1k knockout animal, and by removing ppm1k systemically, what they have is a more inhibited form of BCKDH, and you see that the levels of branched-chain keto acids within the heart accumulate in this setting. And by echocardiography, they're able to demonstrate that this leads to, even in the sham setting, impairments in cardiac function, and in a transaortic constriction model, exacerbation of the heart failure phenotype. They also treated mice with BT2, which is another systemic modulation of branched-chain keto acid levels. And by giving BT2, they inhibit BDK, the BCKDH kinase, and thereby reactivate it systemically. And we and others have shown that this is a terrific way to lower branched-chain keto acids. And by doing this, they were also able to prevent the effects of TAC on cardiac function. And so here we can just see the fractional shortening quantified, where there's a reduction in fractional shortening as one would expect with TAC. And this is abrogated to a large extent with BT2 treatment, which kept the branched-chain keto acids low in those animals. And this is just the left ventricular dimension in systole. And you see a similar abrogation of the negative effects of transaortic constriction. So this suggests that exposure to high keto acids or an inability to oxidize them potentially within the heart may provoke functional cardiac impairments. However, since both of these manipulations were systemic in nature, it's really hard to determine whether it was indeed intrinsic effects within the heart due to intrinsic downregulation of oxidation or due just to high exposition to branched-chain keto acids. And so we felt it was really important to map the fate of branched-chain keto acids in a healthy heart to kind of get an insight into what might actually in fact be perturbed. And so the way we went about doing this is by employing a Langendorf perfusion system with stable isotope labeling of the keto acid from valine, keto isovalerate. We also did similar experiments with the keto acid from leucine, which yielded similar results. The neat thing about keto isovalerate is there is a downstream metabolite that's unique to this pathway, 3-HIB, identified by Zoltarani's group and Chol-Sun Yang, which isn't CoA labeled and can promote fatty acid entry, but can also be exported from the pathway. So we were able to measure labeling not only in KIV, but also in valine and 3-HIB, and also we looked at both succinate and citrate labeling as measures of entry into the TCA cycle, since valine can enter presumably through succinyl-CoA. So when we do this in healthy hearts from Wistar rats, and we also quantitate the pool size and the labeling, we can get a measure of the total production of these intermediates, or total label concentration. And what you can see is actually, rather than branched-chain keto acids in a healthy heart being driven towards oxidation into the TCA cycle, which would be measured by labeling of citrate and succinate, and also 3-HIB, the actual major fate is this reverse transamination, this conversion back to valine. So you're getting an amine group added back. And this had not been described before as a major fate, and so in the heart. And so we wondered whether, if we were to increase activity of BCKDH, we could tilt the distribution of the carbon across the pathway. And so we do find that when we use BT2 to inhibit BDK and activate BCKDH, that there is increased flux or increased labeling of downstream intermediates past BCKDH, which include 3-HIB and also citrate and succinate. But the major fate, being valine, is not drastically affected by this maneuver, which seems to suggest that some of the keto acid is being rapidly reaminated to valine and then sequestered away from the mitochondria and BCKDH. We next used a small molecule inhibitor of the transaminase to demonstrate that this is BCAT dependent. And this is a molecule that inhibits both isoforms of BCAT. There's a cytosolic and mitochondrial form. And when we use this inhibitor, you can see that there is a strong effect to lower label incorporation into the valine pool. And now valine is no longer the major fate of these keto acid-derived carbons. And in fact, 3-Hib, the molecule downstream of BCKDH, begins to become a more important fate. However, very few of those carbons end up going through to citrate and succinate. So this paints a picture wherein the major fate within an isolated healthy heart is not oxidation per se, suggesting that the downregulation of the oxidative machinery within an unhealthy heart potentially isn't the driver of branched-chain keto acid accumulation. And it's maybe more so exposure. But to validate that this reamination effect is, in fact, not just an artifact of the isolated perfusion setting, we moved into, hold on, one last piece of information. We've also measured the concentration of label within the perfusate, and what you can see is a portion of this valine that's being produced from reamination of the branched-chain keto acid within the heart is actually exported from the heart and enters the perfusate. And this is similarly affected by the different compounds. Here, BT2 actually has an effect on the quantity that finds its way into the perfusate, with the inhibitor of the BCAT having the strongest effect. So we moved in vivo and we took healthy Wistar rats and we gave them a bolus of 13C labeled KIV. And so all the carbons are labeled in the KIV. It's the same substrate we used in the isolated hearts. And what you can see, and we took plasma over a time course here. And what you can see is that there's a very rapid reduction in KIV in circulation in the vehicle-treated animals. And this corresponds to a rapid increase in label within the valine pool in the plasma. In fact, within 10 minutes, 40% of the valine in plasma is actually labeled from the KIV, which suggests that this is a significant fate in vivo at a systemic level. And importantly, if you inhibit BCAT, you slow the disappearance of the labeled KIV from plasma, and you abrogate the appearance of labeled valine. So suggesting, of course, that it's BCAT dependent, as one might expect. So which tissues are the major sources or potential major sources of valine produced or branched-chain amino acids produced from the branched-chain keto acids? It appears, in fact, that only heart and gastroc have higher percent labeling of their valine pool than the plasma, suggesting that other tissues such as liver and kidney are not contributing to this reamination. In fact, the heart was the site that we measured of highest labeling, suggesting it's probably the predominant site, in addition to skeletal muscle, for producing branched-chain amino acids from their keto acids. So we have this thing where the heart is doing something unique. It's preferentially reaminating the keto acids rather than oxidizing them, and we wanted to understand why that might be. And so at first we felt that a simple explanation might be a differential expression of the cytosolic versus mitochondrial isoforms of the branched-chain aminotransferase enzyme BCAT1 and 2, respectively. And although there was a trend for a correlation with the cytosolic form, there was nothing really significant among these suggesting that that would be driving the tissue-specific differences in the amount of reamination happening. However, Shingo Kajimura recently identified within his brown fat studies a novel mitochondrial transporter of the branched chain amino acids, which allows the branched chain amino acids to traverse the mitochondrial membrane and go from the cytosol to the mitochondria. And this solute carries SLC25A44. And when we looked at the expression of this novel solute carrier across the different tissues where we measured the accumulation of reaminated valine, we see that there's a very tight correlation between the expression of the mitochondrial importer of the branched chain amino acids and the accumulation of reaminated branched chain amino acids, suggesting that those keto acids that are rapidly reaminated may indeed be sequestered away from the mitochondria and further oxidation because there's low expression of the importer that will get them back from the cytosol into the mitochondria. And so to test this hypothesis, we worked with Charlie Gobach's group at Duke and Jackie Wellacre in our group took on this experiment where we made AAV9 constructs expressing either controlled GFP or the solute carrier, and we injected them by tail vein, and then we performed the same isolated perfusion studies in the hearts of these animals that we performed prior with the KIV in perfusion. And you can see that there was a nice overexpression of SLC25A44 with the AAV. And this resulted in a 50% reduction in the accumulation of reaminated valine within these hearts. So these data suggest that it may be that the low expression of the solute carrier leads to sequestration of reaminated valine within the heart and away from the mitochondria, and that can be overturned by increasing expression. And so this may be a novel therapeutic pathway or a means to lower the accumulation of branched chain keto acids or promote their oxidation within the heart. And we've got some studies ongoing to test those hypotheses. So what is the consequence within the heart of exposing the heart to obese or in high levels of branched chain keto acids that are within the physiologic range? And so there have been other studies where people have looked at the effects of branched chain keto acid, and often these studies are using the hundreds of micromolar doses of branched chain keto acid, which don't really occur in vivo and definitely not the levels we're seeing in plasma. In fact, when we go from lean to obese levels, we're going from a range of 10 to 12 micromoles up to maybe 20 or 30 micromoles, depending on the keto acid. And so we modeled perfusions based on levels that we measured in our lean animals and humans and those within obese plasma, and looked at the effect on protein synthesis. And we measured protein synthesis using a pure myosin exposure that's called the sunset technique. And pure myosin will be incorporated into newly formed proteins because it looks like a T RNA analog. And so you can then use an anti-pure myosin antibody in blood, and you get a non-quantitative readout of changes in protein synthesis rates. And so what you can see here is when you go from having no branched chain keto acids in the perfusion buffer to the levels found in lean animals, there's no effect at all on protein synthesis. So having low exposure or healthy exposure of branched chain keto acids really doesn't impact this pathway in healthy hearts. However, if you take the levels that are found within an obese animal, which are only double those found in lean, you have a rapid doubling of pure myosin incorporation into proteins, suggesting that simply going from lean levels to obese levels of branched chain keto acids is sufficient to increase protein synthesis. And we think this could be important in the setting of diabetes and obesity-related cardiomyopathy, where you have an increase in protein synthesis and leading to unhealthy cardiac hypertrophy. We looked at the signaling here, and we see an increase in phosphorylation of the translational repressor 4-ABP1, which is known to inhibit 4-ABP1 and lead to increased translation. Not with the lean branched chain keto acid levels, but only with the obese levels. which matches what we saw with pure myosin incorporation. So we think that there's evidence that this reverse transamination of branched-chain keto acids and sequestration could be leading to increased activation potential of the mTOR pathway, leading to increased phosphorylation of 4-BP1 and increased protein translation that in the long term may have unhealthy consequences and contribute to cardiac hypertrophy. On that note, what do I want to say there? Yeah, so it's worth mentioning that these incubations were only 30 minutes long. So all this had happened within 30 minutes of exposure to these levels of keto acids. So we were curious about what other networks exposure to branched-chain keto acids might be regulating within the heart, and to determine if there's any kind of coordinated signaling events that might be taking place. And since this effect in protein synthesis happens so quickly, we felt the best way to look at this would be through phosphoproteomics. So we employed tandem mass tag-based phosphoproteomics analysis with the Duke phosphoproteomics core. And what we compared was the high branched-chain keto acid exposed hearts to those exposed to the low levels, not the no level, but the low lean level of branched-chain keto acids. And in fact, there were multiple phosphopeptides that were changed within this 30-minute exposure. We're looking at about 118 phosphopeptides that were downregulated and close to 50 that were upregulated when we used the cutoff of p equals 0.01 or less, and with a twofold change. And if you look at the gene ontology enrichment for those upregulated presented in red and those that are downregulated presented in blue, you can see that a lot of these phosphopeptides are within the cytoskeleton, the actin cytoskeleton related to the actin filament bundle. And within the upregulated, there was also a cluster of phosphopeptides that were related to muscle cell action potential, including the ryanodine receptor and some sodium pumps channels. So this may suggest that exposure to branched-chain keto acids is not just affecting protein translation, but it may also be impacting action potentials within the heart and contractility. Looking within these data for specific pathways of interest, what we identified was that within the downregulated phosphosites, there was the regulatory phosphosite for the p38 MAP kinase was one of the most modulated sites, the tyrosine 182 and threonine 180. And there's also phosphorylation changes on MAPCAP kinase 2, which is a downstream protein downstream of p38 signaling, as well as STAT1 and other sites regulated by p38 MAP kinase. This suggests that branched-chain keto acid exposure might be reducing signaling through the p38 pathway within the heart. And within the upregulated phosphosites, we see proteins associated with actin and myofibril polymerization, including the CAP-ZIP protein, CAP-Z interacting protein, which is also known as RCSD1. And this particular phosphorylation site, as well as a number of others within these upregulated phosphosites, are known ERK-regulated phosphosites. And so when we look at the phosphosite motifs of these regulated, downregulated phosphosites and the upregulated phosphosites, we do appear to see a conserved motif, or at least a kind of consensus sites. And the one we see for downregulated is consistent with decreased p38 MAP kinase activity. And the upregulated phosphosite is definitely consistent with increased ERK signaling, which is consistent with some of the phosphopeptides we see within that list, and also changes in things like ARAF and RAPGEF and cortactin phosphorylation. So these data suggest that the branched-chain keto acids might be moving beyond simple mTOR signaling and also causing a shift in the balance between p38 MAP kinase signaling and ERK signaling. And both of those pathways have been associated with changes in cardiac hypertrophy. Within knockout mouse models, the absence of p38 MAP kinase has promoted increased hypertrophy. And so, although that's a controversial area, these are giving us some new and important clues into how branched-chain keto acids might be eliciting some of their effects. We also mined the data for potential proteins within the mTOR pathway to look at what's going on with 4-ABP phosphorylation. And indeed, a phosphosite on Raptor was increased. And this phosphosite, serine 859, is actually known to be regulated by ERK and a means to regulate mTOR activity towards 4-ABP1, further supporting this model. Moving beyond the motifs, we also looked at the interactome among phosphoproteins that were upregulated or downregulated, and in line with the fact that there's not so many upregulated phosphosites, only 2 interactome nodes were identified, and one of them is the one associated potentially with contractility and action potential. Here's the ryanodine receptor, and some of the sodium transporters. And we also see another node, which is supervillain and cortactin. These are associated with actin polymerization and myogenesis. Supervillain is one of the first complexes formed during myogenesis, which suggests that there may be the phosphorylation is maybe coordinated towards driving hypertrophy, as we postulate. And we also see, when we look at the downregulated phosphoproteome, actually quite strong kind of interactomes appearing, and a number of different nodes that seem to be regulating, suggesting that the biology that branched-chain keto acids are hitting may go beyond simple p38 MAP kinase and ERK signaling and mTOR signaling. We also see, aligned with that, MAP kinase 14 or p38 MAP kinase jumping out as one node, but there's also 26X proteasome has multiple components that are jumping out at us. And then this AMP to IMP regulation, this is AMP deaminase and adenylate succinate synthetase ligase, which is convert AMP to IMP and vice versa. And we also see hints of networks related to lipid storage. Here's hormone-sensitive lipase and perilipin 5. So these data are going to inform us, I think, in the future about what nodes branched-chain keto acids might be affecting. But it looks like there's a strongly coordinated response to exposure to branched-chain keto acids, and that they may be underlying important facets of the pathobiology of cardiac myopathy. So to summarize what I've just shared with you, we used a tracing approach using 13C KIV in isolated perfused hearts to identify that reamination of the keto acids to their branched-chain amino acids is actually the major fate of keto acids when they come into the heart. And in fact, there's not a lot of oxidation that takes place, except if you activate BCKDH by inhibiting the kinase. And even in that sense, very little makes it to the TCA cycle. We think that the reaminated amino acids are sequestered away from the mitochondria due to low levels of expression of the mitochondrial solute carrier for the branched-chain amino acids, SLC25A44, within heart, and that's a tissue-specific expression pattern. And so when you expose hearts to levels of branched-chain keto acid that are found in pathologic conditions such as obesity and insulin resistance, what happens is you get an increase in protein synthesis that seems to be corresponding to an increase in phosphorylation of 4-BP1, but also potentially effects through suppression of the p38 MAP kinase pathway and possibly through activation of ERK and changes in phosphorylation across a number of proteins related to the cytoskeleton. We hypothesize that this could be involved in cardiac myopathy, and these are where our future studies are headed. So before I end, I just want to do a shout out to the wonderful members of our lab that have been involved in this work. A lot of the perfusion studies that I talked to you about today were carried out by Bridget, cardiology fellow who's in our lab, Bridget Christopher, and also Jackie Waleko, a new postdoctoral fellow in our lab, and in conjunction with Guofeng Zhang and Scott Crown, who run the stable isotope tracing metabolomics within the DMPI. We also have important collaborations from Shingo Kajimura and Takeshi Yamashiro, and Charlie Gershbach and Adrian Oliver at Duke. And our work is funded by a grant from the American Diabetes Association to myself, as well as from Lily. And so I hope I haven't taken up too much of the time. Thank you for listening.
Okay, great, Phil. Thank you. This was a great presentation and a lot of unpublished data. So we thank you again for that. If anyone has a question for Phil, please use the raise your hand function. I'll start with the first question. So Phil, you also mentioned this, but the role of P38 is controversial in heart failure, right? Whether it gets activated, but I think MK2 is more. I know a paper from Yibin's lab, I guess, where they show that MK2 inhibition protects from heart failure. So since your experimental approach was acute effects, and that there you see a lowering of MK2, did you check like chronic effects of chronic branch chain amino acid effects on those phosphoproteins, or are you planning to?
That's a terrific question. That's exactly where we're headed next. The isolated perfusion system is terrific for acute exposures, but it's very hard to keep the heart happy and beating for much longer than 45 minutes. So we've got some nice genetic models that we think are going to allow us to achieve increased exposure of the heart to elevated levels of branched-chain keto acids. So we've made the EDK floxed animal. And so we're going to be able to study those animals where we've knocked it out in liver and see whether long-term exposure, what it does, and then interrogate these same pathways. So I think that's important. Yeah.
All right, great. So I see that Ebru has a question, but we can, yeah, Ebru, would you like to go? Because I first saw your hand.
Well, okay. So thank you, Phil, for this talk. Thank you for accepting to talk. And I have a question. So the phosphoproteome that you showed us represents the heart being exposed to the branched-chain amino acids. But what would happen if the phosphoproteome was different in an obese heart, for example?
So if we, would the changes be the same in an obese heart exposed to branched-chain keto acids versus a lean heart? Yeah, these I think are things we're going to have to kind of move forward and validate. I think what we wanted to do is begin with a healthy heart and ask, does just exposing them to this feature of obesity without all the other confounding factors have any significant impact? And I was not expecting such a broad effect on the phosphoproteome, and something that seems to be, and I definitely was surprised by the effect we had on protein synthesis. So these are things we're going to need to explore. Also, in the heart failure models where you have a downregulation of the pathway, are these same pathways activated by exposure to the keto acids, and is it exacerbated? And so these are kind of the things that we need to move on to next.
Mm-hmm.
Yeah.
And I think in your in vivo models, you'll be able to do that.
Yes.
Yeah. Thank you.
Okay, we have two more questions. Thomas, please go ahead.
Thank you. Nice talk, Phil. Thomas Dalhousie. What I know, it's a philosophical question. Heart uses fatty acids in a more dominant way. So what's the impact of fatty acids on branched-chain keto acid metabolism? That was my first question. I mean, what is this data telling us? And second thing is, have you in your tracing studies, did you use competing substrates while doing those experiments, or was it only the valine and the keto valine?
And so within the perfusion buffer, there are palmitate, there's glucose, there's oleate. So there's the general substrates that would always be there, and then we've added on top of that just the KIV or the KIC, or in the case of the branched chain keto acid kind of cluster exposure, all three branched chain keto acids. We haven't gotten around to looking at what the impact is on substrate preference. We've done a prior study, which was published in 2019 in AJP, where we looked at the effect of branched chain amino acid restriction on substrate preference. And we can do that by using differentially labeled palmitate versus glucose, and then looking at the ratios of carbon incorporation or carbon patterns at the acetyl-CoA level. And we did see that when we restricted branched chain amino acid supply to a Zucker fatty heart, that we actually promoted fatty acid oxidation and reduced glucose utilization of those hearts and led to a reduction in triglyceride accumulation. And this is something that we want to see whether or not we get a similar effect or an inverse effect when we do branched chain keto acid exposure. So that's another set of studies we have kind of in the pipe, where we're going to be treating with the branched chain keto acids, and instead of looking at protein synthesis, looking at substrate preferences.
Also, just one last thing. When you do the tracing studies, could you have a choice of doing it in a fed versus fasted heart? Have you compared how the data looks like, and if there is a nutritional modulation of the heart that is isolated from?
You could choose to do that. We haven't done that. We take them from fasted animals, I believe, and so that's another thing that needs to be examined. I guess it's something we can rest to be examined, is does this effect look the same across those conditions? It's a little bit different from looking at signaling within the animal at sacrifice, in that they're within a buffer, a perfusion buffer. When they're isolated, they're equilibrated within that buffer, and then we look at that phase. So I think the only thing we'd probably pick up there that was left over from the status of the animal would be gene expression changes that were long-standing. But anything that was acute, I think, would probably be washed out during the washing phase. So, but yeah, it's definitely worth looking at in our next studies.
Thank you.
Okay, I think we have one more question. Yes, Mabruka. Yeah, you can unmute.
Yeah, hi. My internet connection is pretty much weak today, so I do apologize if I got disconnected. So your talk is pretty much interesting. So I'm highly interested in the translational aspect of your research, and your phosphoproteomic data really shows some very complex interactions. So have you seen like a similar changes in heart failure patients' heart? So in the heart of patients have heart failure. A second question is, the pathway looks pretty much complex. So are you gonna target the upstream of branched chain fatty acids, or you're gonna look for downstreams of where the B38 is B38 and the MAP kinase ERK signaling is being different? Thank you.
So I'll start with the second part of the question, because I think that's something that we've thought a lot about, and we think that it's likely that the changes that we're seeing or the associations that are being observed with elevated branched chain keto acids and heart failure, et cetera, are due to increased exposure of the keto acids to the heart and their accumulation therein. So this opens the door to targeting oxidation at other sites as a means to lower exposure of the heart. And so you can imagine you could target BCKAH in the liver to lead to lower levels of keto acids within the circulation and kind of a rescue on the heart. I think if we can keep keto acids at a lean, healthy level, we can potentially override that. And I think that's the easiest place to target. but I may be proven wrong about that. And we're trying to test those models at the moment. And then about the translational aspect, I think in that paper in Circulation from Yibin Wang's group, they demonstrated quite nicely that the branched-chain keto acid accumulation is not just a feature in mice, but is also a feature in humans, and I probably skipped over that. So I think that, translationally, I think it's relevant. We have some nice new data showing that the feature that distinguishes between obese individuals with non-alcoholic fatty liver disease or not, is not actually the branched-chain amino acids themselves, but their keto acids and the ratio of the keto acids to the amino acids. So, they're a little bit more challenging to measure. So I think, we kind of think a lot about branched-chain amino acids, but I think the keto acids may have really important functions and something that we can go after more specifically.
Thank you.
Okay, all right, great. Thank you for all the questions, and thanks, Phil, again for an outstanding talk. And then Ebru is going to introduce our second speaker.
Well, thank you, Phil, again for joining us with your seminar today. I want to introduce you to Alessandro Bartolomucci, who's a faculty at University of Minnesota. He's been there for 10 years, so I can't believe that. It's been a long time already. He's a native of Italy. Most of his career was built in Parma. So, and I think he's been working on stress for 2 decades. So he's a real expert on how stress impacts metabolism, which we are very stressed right now. So we need to listen carefully. It's all yours, Alessandro. Welcome.
Hi, Ebru. Thank you very much again for inviting me and for your heroic effort of keeping this cardiometabolic series going. Let me see if this is going to work. All right. Do you see my presentation and such? Let's see if I can. I hate to see you all on the side, but okay, I'm going to live with that. Okay, thank you very much again. So I would kind of switch gear quite a lot.
Alessandro, it's not in presentation mode, just so you know.
It's not?
Yeah, not full.
It is in my.
We see a big slide and a small.
Is it now?
Perfect.
Okay, that's weird because I see now this in a different screen. Okay, well, anyway. So if you see myself pointing on the other direction, it's not because I'm ignoring you, but because for some reason my screen is projecting the second screen. Anyway, so what I was doing, I was saying is that today what I will do is to summarize some of the work that we've been doing, as Ebru said, in the last 20 years or so. First in Europe, in my lab in Parma, in different other labs in Europe, and then when I moved to the University of Minnesota about 10 years ago now. And I will try to link a couple of aspects that we've been investigating quite a lot over the past few years, and which has to do really with the impact of social stress on cardiometabolic diseases. And when I was kind of building this talk, I kind of ended up into this good, bad, and ugly concept, and I was really forced to recognize Ennio Morricone, who died a couple of I guess weeks ago. Nobody paid much attention because of the COVID, but anyway, so the subtitle is kind of forced to recognize his work, which has nothing to do, of course, with what I'm going to present today. A short introduction of what we do in the lab, mostly 3 major research projects. One has to do with thermogenesis, brown adipose tissue function. The other has to do with stress, cardiometabolic, and disease of aging. And so I will kind of merge these 2 in the talk. And then we also have a large project on neuropeptides called TLCOP21 and how this affects obesity. And I'm not going to really touch upon this at all today. So what is stress? So Ebru said we are all stressed, etc. But our definition of stress is slightly different, actually. And I will change a little bit the perspective. So the stress physiology that we know a lot really has ancient roots. It really started with Walter Cannon, with Claude Bernard, and many other major physiologists. And this is really the physiological reaction, which is the prerequisite or required for for behavior, for any normal function. And a couple of years ago, we wrote a review as a kind of review point to create a revised terminology. And rather than using this broad terminology, leave this to physiological homeostasis and not call this physiological adaptation stress. If this is not stress, then what we suggest stress should be reserved for, essentially for potentially life-threatening conditions, which are characterized based on a number of data by unpredictability and loss of control. And you know, this will take quite some time to describe all the evidences and what we mean exactly. And a stressor, so the condition eliciting the stress response, is not just a normal life event, but is rather a condition which can exceed the regular regulatory range and capacity of an organism. So it's a rather narrow and, if you want, a pathophysiological view on what stress is. And there are a lot of evidences or support to this conclusion. And when we look into stress in this revised term, that's where a lot of the pathologies come out. We know that the stress is a major risk factor when restricted to really these conditions, to a number of diseases. And now there is a lot of discussion, and we are doing some little work at the moment, even on how this can increase the risk for COVID-19, at least for the major disease conditions, or the major, the worst outcome for people affected. Again, building on this concept, we know that sustained life stress and the measure of low socioeconomic status, which is a very complex construct by epidemiologists and psychologists, but nevertheless encompasses a lot of the what we consider stress. We see a lot of the negative effects on lifespan and diseases. For example, in this very impressive study published by Silvia Stringini a few years ago, the hazard ratio of low socioeconomic status was as high or higher than hypertension and obesity. So not a minor effect, a major effect at the population level. This was several million people screened in an epidemiological study. In more controlled studies, the classical WIDO2 study, lower survival in people with high stress, similar in non-human primates, similar in rodents. And I will touch upon this at the end. And this year we published a review on summarizes some of these cross-species comparison and the social determinants of health, and why social will become clear in a second. So how do we work on mice in my lab, mice, cells, and mechanistic studies? How can we study this severe social stress in rodents? The model we have been using and developing over many years very briefly works this way. You have two male mice. For complex reasons, this works mostly or only really in male mice. You have one animal that will be introduced into the cage of an aggressive conspecific. And very often the intruder would be a C57-like strain. We are forced by geneticists to use the strain. Nevertheless, this is our strain of election nowadays. And then the resident will be this CD1 male, which is highly aggressive outbred strain. The two will be facing each other. There will be an aggressive interaction. And then, very importantly, we leave this animal in sensory contact for the rest of the day, and really for as long as you want. Most of the studies are going to show today going to be in the range of weeks. There will be also a lifespan study at the end of the talk. And out of this confrontation, one of the two animals, almost invariably the C57 in this example, will become subordinate, will be an animal of low social rank. And you can really use this paradigm for any strain you want. If you are interested in studying social dominance, then send me an email, because the situation is much more complicated and we are much more limited in what we can do. So why this model and what we know of this model? This is just a summary table of as Ebru pointed out, about 20 years of work. We have been measuring a number of physiological functions, and kind of a punchline of this story is that the subordinate animal, and again, the subordinate animal in this chronic stress will show major physiological changes, major neuroendocrine changes, HPA axis and catecholamines, cardiovascular imbalance, metabolic and behavioral changes. Two important points, and I'm not really showing much of the data here, but these physiological changes are very remarkable and much higher than you can get with any other stress model that has been tested so far. And even more importantly, the physiological changes are almost exclusive to subordinate animals. There is very little going on in the dominant, with notable exception that I don't have time to discuss today. So really is a unique physiological response. And so what I would like to discuss with you today is really two aspects that we've been investigating. One has to do with the role of the brown adipose tissue, or in any case, the impact of stress on obesity, and the second of stress as a risk factor for cardiovascular diseases. Let's start with the metabolism, obesity, brown adipose tissue first. One of the, for the psychiatrists, psychologists in the audience, one of the major factors or evidences in this field is that when you expose organism, really human animal model, to stress, you can really have one of two outcomes. They can become obese, or they can actually be resistant to obesity, or actually lose weight. And interestingly enough, this is not univocally associated with hypophagia or hyperphagia. There are conditions of hyperphagia actually associated with obesity resistance. And the field has always been really split on this dichotomic effect without a clear answer. A few years ago, based on some of the data I'm going to show you today, we focused a lot and we pointed our attention to the brown adipose tissue and how the brown adipose tissue can play a role here. So what is the brown adipose tissue? Well, this is not coming as it was supposed to be. Okay, nevertheless, it's really a mammalian adaptation. And a few weeks ago, we had a wonderful set of talk by Claudio Villanueva and Alexander Baltelt. And I'm not going to repeat all the beautiful cell physiology and biochemistry that they highlighted. But I want to point to some physiological role of the brown adipose tissue, which is important for this talk. First of all, the brown adipose tissue, or differentiation, amount, and really function, is inversely associated with body size. And there is a striking difference, for example, between rodents and humans in terms of the recruitment of this organ. And why is this? One of the reasons is that the brown adipose tissue subserves as a function the adaptive thermogenesis. And when you have a large organism with a lot of additional body to increase or to produce thermogenesis and to avoid heat dissipation, you don't need a lot of extra calories to produce temperature, which is required to keep us at our core body temperature. And so what happens is this interesting curve. If you are for a species within a thermoneutral zone, your adaptive basal metabolism rate is very stable. As you can see in this wonderful work of Mark Reitman, accounts for most of the total thermogenesis. But as soon as you get away and in below the low critical temperature, you need more energy expenditure to produce the extra heat to keep your core body temperature stable. And which organs are producing that is mostly brown adipose tissue and cold adaptive thermogenesis, although obviously the skeletal muscle and other organs clearly play a role. But this is very important. And if you look morphologically, that is reflected in the morphology. The brown adipose tissue at thermoneutrality, this is an evidence from a mouse, is completely different. The adipocytes looks much like white adipocytes, circle lipid droplet, very few UCP1, and very importantly, something that people often forget, there is a much less sympathetic innervation in these conditions. Here highlighted with these red arrows. This is tyrosine hydroxylase staining. This is the first important point of the brown adipose tissue. The others are, UCP1 is not leaky. It's not just a protein that discharges the proton gradient continuously if it's not needed. The brown adipose tissue is a very costly organ, and it requires an input. Where is this input coming from? Mostly from the brain. The brown adipose tissue is kept under constant inhibition by an inhibitory signal in the brain, mostly the dorsomedial hypothalamus and raphe pallidus, identified by Sean Morrison in many years ago. And so this is kept constantly inhibited. And when, for example, in this example, the skin is cooled down, then there is a rapid discharge of sympathetic nerves and a rapid activation of the brown adipose tissue. So this is a very important, and if you combine the work of Barbara Cannon, Jan Edergaard, and Sean Morrison, you really have this picture of an organ which is kept inhibited unless it's needed for something. The other important point is the role of the sympathetic nerves. Sympathetic nerves are known to be necessary for brown adipose tissue recruitment. Okay, if you back in 1954, they did surgical unilateral denervation, and if you cut the nerves to the brown adipose tissue, you end up in a morphology like this. Okay, clearly non-brown adipocytes, white-like adipocytes. And consistently, if you generate an animal without the 3 beta-adrenergic receptor, this is the so-called beta-less mice from Brad Lowell, you end up with a brown adipose tissue which looks much like the white adipose tissue. And this is an image of the same strain we collected using the 7 Tesla MRI. As you can see, the brown adipose tissue essentially disappears. The fifth point, if you want, the sympathetic nerves are sufficient for brown adipose tissue activation. And this is just an example from a couple of experiments we published recently. We transfected sympathetic nerves, which are rhodopsin 2, using either viral viruses, AB6 in this case, or a genetic model. And then we used optogenetic stimulation to activate the nerves and not the brown adipose tissue. And the result, as you can see here, was a clear thermogenesis, both of the brown adipose tissue and the core body temperature, which was absent in a number of control animals we used. And similar data were actually published a couple of years earlier by another group. So to summarize the brown adipose tissue physiology, all these conditions, minimal recruitment, the UCP1 not being leaky, the UCP1 requiring disinhibition really explain why the brown adipose tissue and UCP1 mediated thermogenesis is very minimal at thermoneutrality. Okay, it's not needed, it's not required, and is a very expensive organ from a metabolic standpoint. And then the requirement and necessary and sufficient role of sympathetic nerve then kind of suggested to us a different question. That is, can we interrogate condition under which stimulating the sympathetic nerves, we can actually recruit the brown adipose tissue at thermoneutrality? Okay, and I will explain you why in a second. This goes back to a critical question and the link to the first part of the talk. Which are the stimuli that will elicit a disinhibition of that inhibitory circuit? We know cold, of course. Cold is the classical stimulus used in many labs, and including our. And then there are a different approach, which is very important to keep in mind, that is not equivalent to cold. If you use a beta agonist, you're going to stimulate the adipocytes, but you're not mimicking, you're not activating the sympathetic nerves. So you're only activating downstream of that sympathetic activation. And the third stimuli, is something that we and others have been identified, is actually stress. And that's the link to the rest of the story. There is beautiful work done by Katsuhiro Nakamura, which was a former postdoc with Sean Morrison, who identified basically almost the entire, at this point, circuit from the frontal cortex, prefrontal cortex, and hypothalamus, leading to psychosocial stress, social defeat, a very much similar model than we are using, although in an acute setting, in eliciting the disinhibition of that pathway, thermogenesis of the brown adipose tissue, and pretty much all the story you are used to in terms of cold. And in our work, what we did was more physiological. So we have animals in the intercalorimetry, we measure energy expenditure before and after the social defeat. We cannot do it during, because of course we need only one animal in calorimetry. And so what you see is this very massive increase in VO2, for example, right after the social defeat. This is 10 minutes after, which is completely blocked in the beta-less mice. So beta-less mice don't have beta-adrenergic receptor. Their thermogenic response is gone in response to this social defeat. And so the first question that we're trying to ask really is, can the housing temperature, keeping in mind that we are using here housing temperature more as a way of testing brown adipose tissue function. So we're going to use 22 Celsius, which is the standard room temperature in which we know the brown adipose tissue is very much recruited in mice, and 30 Celsius as a way of minimizing adaptive thermogenesis and brown adipose tissue recruitment in rodents. And then the animal were exposed to stress for 5 weeks in condition of high-fat diet. Okay, and what happens at room temperature is that the animal become hyperphagic, they gain weight, their fat mass increases, and they become glucose intolerant. Okay, I'm not showing a number of other data. But what happens when you do the exact same experiment at thermoneutrality? Behaviorally, neuroendocrine response, all are virtually identical. The animals are still hyperphagic, The absolute level is actually much lower because animals are at thermal neutrality, but they are still becoming hyperphagic. But yet they resist obesity, they don't increase fat mass, and they remain euglycemic. How is that possible? Remember that these animals are becoming hyperphagic. So again, that points to that difference. The major surprise when we were looking at the brown adipose tissue of this animal, consistent with this pro-obesogenic effect at room temperature, we don't really see much in terms of morphology, UCP1. If anything, we see a whitening of the brown adipose tissue. But when you look into the animal at 30 Celsius thermal neutrality with high-fat diet, what we saw was a massive increase in UCP1 and a brown adipose tissue, for the expert in the audience, which looks almost indistinguishable from a wild-type control at room temperature. This is pretty impressive because it's browning at thermal neutrality, something that we called a little emphatically butt browning. It's not just the brown adipose tissue. Sympathetic nerves follow the same trend, massive increase in sympathetic reinnervation, if you want, of this organ induced by stress at thermal neutrality. And then that led to the follow-up question, if you want, which is still unpublished these days. That is, if sympathetic nerves are increased and the brown adipose tissue is recruited, can we prevent this effect by doing a surgical denervation of the brown adipose tissue? What we did was a pretty heroic experiment. Mice were acclimatized at thermal neutrality, surgical denervation, 5 weeks of chronic psychosocial stress, TH essentially gone in the brown adipose tissue. And the question is, will this animal without nerves connecting the brain to the brown adipose tissue be able to recruit and have the beneficial, if you want, effect of stress at thermal neutrality, which is summarized here? And the answer, of course, is no. When you cut the nerves, the animal is still mildly hyperphagic, much less than normal, and this can be very revealing of the role of sensory nerves in this context. But in any case, they don't increase fat mass, they essentially increase body weight, they don't lose weight, and UCP1 doesn't go up. So the sympathetic nerves are very required for this stress-induced browning at thermal neutrality and the anti-obesity effect. So the first part of this talk essentially points to stress to be an alternative stimulus for this brown adipose tissue sympathetic activation. And essentially is very selective, and I didn't show the data, it's all in the paper, but we have excluded a major role of the subcutaneous or the perigonadal white adipose tissue. And this is not unexpected because the sympathetic innervation is very local, it's very specific to each organ. It's not just a random activation of all the sympathetic nerves. And so this brown adipose tissue recruitment at thermal neutrality appears also to be sufficient to limit diet-induced obesity. Does the good in my title. But obviously, we are not interested in going around and suggesting people to be overstressed to prevent obesity, because unfortunately, as we know, very often stress is linked to binge eating, to hyperphagia. It actually results in obesity unless you have other mechanisms compensating for this. But the question really is, can we use this mechanism to identify novel approaches for obesity? And this goes back to the kind of the simplistic view that a lot of us have and use in a number of papers. That is, sympathetic nerves secrete norepinephrine, which activates beta-adrenergic receptor, and then the cascade with free fatty acids and UCP1 and HIT. If this was the full story, then beta-less mice, which lacks beta-adrenergic receptor, should be resistant to stress-induced obesity. If all the story that I've been telling you so far is correct, stress elicits sympathetic activation, and it's all mediated by beta-adrenergic receptor, they should not respond. And one of the key points of the paper that we published a few years ago now is that these beta-less mice not only are not resistant, but essentially they recruit the brown adipose tissue with normal brown adipocytes expressing UCP1 and virtually all the physiological conditions that you can think of this organ in the condition of stress in the brown adipose tissue. Okay, and they do this in presence of increased sympathetic innervation, increased energy expenditure, and resistance to diet-induced obesity. How is this possible? If this is the version that we are used to, how can a beta-less mouse respond to this? Obviously, this led to so many hypotheses that took my lab busy, and anyway, we have so much unpublished work that if somebody's interested in this topic, send me an email and you can come and work on this data for many years and build on this for generations, I guess. But one story that I want to tell you today has to do with one of those factors that we investigated the most. That is this idea that sympathetic nerves are not just secreting norepinephrine. Sympathetic nerves secrete a number of molecules, okay? So forget about this idea of sympathetic norepinephrine. That's only part of the story. Sympathetic nerves produce MPY, they produce ATP, secrete, sorry, not produce, ATP, as well as this TLQP21 that we are investigating. And really, Jeffrey Bernstock is the person that comes to mind, speaking of sympathetic co-transmission. For this to be true, we need to identify something which is expressed in the sympathetic nerves in the brown adipose tissue, which is not norepinephrine, and which has a receptor on the target cell, brown adipocytes, okay? And long story short, our study focused a lot on the ATP as a secreted factor. Why? We have been able to identify the vesicular nucleotide transporter, and this is immunohistochemistry from human brown adipose tissue, in the sympathetic nerves, as well as, interestingly enough, in the brown adipocytes of humans, and almost identical in mice. And VNUT is required for the accumulation of ATP in secretory vesicles, which is required for then doing synaptic transmission. On the other hand, the brown adipocytes are known to express a number of purinergic receptors. And there have been quite a lot of interest recently on adenosine, on the A2A, etc. But for a couple of reasons, we decided actually to focus our attention on the P2X5. Why? P2X5 actually came out in a very elegant paper from Sigrid Ussar when he was a postdoc with Ron Kahn as one of the membrane marker of brown adipocytes. So that was kind of intriguing because there was a pretty selective marker of brown of what we were studying. And second of all, we generated immortalized wild type and brown adipocytes, and then we were treating. Unfortunately, there is not yet any selective P2X5 agonist, but ATP gamma S has been used, and there is some evidence that is relatively more potent, I should say, not super selective for the P2X5. And what is interesting is that ATP gamma S would induce browning, here presented as a UCP1 expression in vitro, both in wild type and beta-less mice. So this appears as a possible link in explaining the physiology that we saw in vivo. And then we generated, and this is a lot of unpublished work, the P2X5 knockout immortalized pre-adipocytes, okay? And this is consistent with our story. When you knock down the P2X5, not complete knockout, but about 50% knockdown, the adipocytes are much less likely to differentiate, and they express much less UCP1. And when you stimulate them with either ATP gamma S or the classical beta-3 agonist CL, 236, blah, blah, what you observe is essentially a complete absence of response in terms of P38 and other pathways. So the cells at this point are less able to respond to stimuli which mimic the sympathetic transmission. And then we move this in vivo. Again, I consider myself more of an in vivo physiology, although I'm using much more cell physiology now these days. But when we treated then obese animals, these are wild-type animals, acclimatized at thermoneutrality and treated chronically for 2 weeks in this case with the CL, the beta-3 agonist, or 2 different doses of ATP gamma S, what we could see is that as expected, and there are a number of works from Mark Reitman, the CL would induce browning, and of course have an anti-obesity effect and lower fat mass, again at thermoneutrality, with increased UCP1. ATP gamma S was actually doing something very similar. And then the question is, can this be mediated by the P2X5 receptor? We obtained the P2X5 knockout from Jianguan Choi at UPenn. And actually, as you can see, the P2X5 is absolutely required for these anti-obesity effects at thermoneutrality, as well as for the decrease in fat mass and browning, which is not shown here. Interestingly enough, this P2X5 knockout also showed blunted response to the beta-3 agonist, which is again consistent with the idea that this co-transmission in this receptor is important for the brown adipose tissue function. So to kind of sum up this part of the story, we have already mentioned these 2 conclusions, but what we are really interested in and what we were able to see by using this model of stress. In this case, we didn't use stress as a way of studying how stress increased the brown adipose tissue, but we really are studying this model as a way of eliciting a selective sympathetic activation of the brown adipose tissue. And in this case, this sympathetic co-transmission is something that we hope can be very exciting and can be a new opportunity for investigating new ways of recruiting and activating the brown adipose tissue at thermal neutrality with potential anti-obesity effects. So in the second part of the talk, which will be much, much shorter, of course, in the interest of time, I want to also share with you some of the physiology of this animal, because right now we're looking at obesity and adipose tissue function, but what's going on in the rest of the body? We spent quite some time by working on cardiovascular function. Many years ago, this was still during my PhD, we were using DSI radiotelemetry to measure heart rate and cardiac function of these animals. And what we noticed in the subordinate animals was a remarkable inactivation of the cardiovascular or the heart function. Animals during the social defeat, the heart rate of this subordinate animal goes up to 850 beats per minute, which is close to the theoretical maximum that the mouse heart can beat. It's a remarkable value. And this is not just acute. This is not just 10 minutes. This is acute measure day 1, day 7, and day 14. But the animal remains tachycardic for days with values about 100 beats per minute higher than normal. More recently, we became interested in blood pressure. And again, using radiotelemetry, we were able to show for the first time, to my knowledge, that not only the animal developed tachycardia, the animal also developed hypertension. Almost immediately as they start from a baseline level with a mean arterial pressure around 100 or so, they start the chronic stress, and then this is radiotelemetry, so this is a live continuous monitoring of cardiovascular function, or blood pressure in this case. The mean arterial pressure goes up with systolic much higher than 130 or so, and then the mean arterial pressure stably over 120 for days. So this is pretty impressive and exciting to me, and we are spending now a lot of time to work on this concept and this idea. And it's also important because it adds social stress as one of the possible models that we can use to study hypertension in humans, which is not trivial because this model is pretty spontaneous. The animal developed this hypertension spontaneously and without any genetic manipulation and whatever. And what I want to share with you today is very quickly a couple of examples, although I'll probably focus only on one in the interest of time. One is essentially, can stress increase the risk factor induced by genetic predisposition to diseases? The genetic model we have been working on for a number of reasons was actually Duchenne muscular dystrophy, which might be familiar to some and not to others. It's a disease caused by a mutation in the dystrophin gene. It's pretty slow progression in humans, but essentially, unfortunately, most of the kids die for cardiorespiratory failure in the third decade. It's a very severe, uncurable disease so far. And the mouse model that we've been using is per se pretty mild phenotype. They don't develop this dramatic age-dependent onset that you see in humans. And this is called the MDX. And we are also using two different strains, which I'll only very briefly allude to today. One essentially is a rescue of dystrophin only in the skeletal muscle. This is called MTB. And the second is a transgenic overexpression, again, only skeletal muscle of utrophin. And this is just the Western blot summarizing this. So we expose this animal to social defeat. And I believe that sometimes the image speaks much more than any words on whatever else. So, no, I went out of presentation mode. So this is this MDX mouse before the social defeat in a white environment, whatever. And this is 10 minutes after the social defeat. Remember, these animals have a pretty normal phenotype under normal condition. They are not shaking. They are not having a dystrophin condition or a dystrophic-like behavior under normal condition. So this was a pretty extreme phenotype that we observed. And that really got us very excited about continuing this project. And the first observation we made years ago was that if you expose a sufficient number and well-powered study to this only 24-hour of social defeat model with 10 minutes of defeat and sensory contact, about 30% of the MDX animal that you can see here will die. 30% in less than 24 hours. And this is completely rescued by skeletal muscle specific expression of eutrophin or dystrophin. But then we extended this to sort of a lifespan, if you want, a couple of weeks in this case study. And this number goes down to almost 80% in 2 days. And then we always have a few surviving animals, which you don't know what they are. Again, dystrophin in the skeletal muscle or eutrophin almost completely rescue this phenotype. So now we have a very strong quantifiable variable, which is survival. And now we want to understand how is this possible? What is the physiological function which goes wrong in these animals in response to stress? First of all, what happens to the heart and skeletal muscle? Stress per se induces quite a robust increase in damage to the skeletal muscle of the heart, both acutely and chronically. It's all in the paper. I'm just quickly going through this. And the damage to the heart, interestingly enough, is not rescued by dystrophin or eutrophin in the skeletal muscle, essentially explaining that there is a dissociation between the mortality, the animals die or are rescued, the MTB and the Fiona, but the heart is still damaged, even if these animals are not dying, at least for these 2 weeks. So what can be the cause of death in this animal if it's, if it's not the heart per se, in terms of the morphology? Can it be an exaggerated stress response? Very quickly, we used again radiotelemetry. And so the original idea was that we would see an exaggerated cardiovascular response. The outcome was pretty different. First of all, if you apply a very mild stress like a scruffing, something you all do pretty much every day by injecting a mouse, okay? You take an animal, you scruff the mouse, you inject the mouse. You think it's an irrelevant event for a mouse. It actually is not. The animal developed hypertension, which lasts quite some time, I can tell you. I don't have all the data here. But this is the normal response in the animal, on the wild type animals in black. But if you look at the MDX, what happens is very strange, unusual. This animal develop hypotension, transient hypotension. The effect is much more remarkable when you do the single social defeat. Once again, all this is rescued by the other 2 strains. And what happens if you then let the animal go past the first couple of minutes in the social defeat? That the MDX animal develop essentially a lethal hypotension rather than hypertension. The wild type animal are becoming hypertensive. This is the level that you see in wild type. The 2 rescued strains are pretty normal, but the MDX essentially collapse. Their blood pressure drops, and the animal that die, this 30% at 24 hours, have very low blood pressure. Okay, so this for the stress physiology in the audience, and we still don't have an answer on what exactly is the mechanism, but it's definitely a support to the idea that it's not just an exaggerated stress response that can kill you. It's also the failure of an adaptive stress response that can kill you. And this is, to my knowledge, one of the most dramatic evidences that this is the case. I will skip on this example, but we have a lot of work going on with Beshai Zordoki on the interaction between doxorubicin, induced cardiotoxicity and stress, and actually Beshai is recruiting a postdoc, so if somebody's interested, shoot him or me an email. But I want to just go to one last point, and then I'm gonna close this presentation. So we know that these animals, these subordinate animals, have a number of risk factors for cardiometabolic diseases, okay? They develop tachycardia, they develop hypertension. I haven't shown you the data, but they develop dyslipidemia, hyperglycemia, glucose intolerance. There are damage to the heart, the myocardial fibrosis, cardiac remodeling. So the question really is, and this will go to the grand finale to honor Ennio Morricone, can this pay a toll on the lifespan? Can this have effect on the long term? Okay, not just an acute physiological change, not just a couple of weeks and some physiological change, but can this persistently change in a negative way the function of this animal, impact lifespan? We extended the study, which normally runs only a couple of weeks, to lifelong. Okay, we continue this experiment, keeping the animal in sensory contact, not the social defeat anymore. This is a very complex experimental protocol, but the data I'm going to show you right now have to do with C57 Black 6 becoming either dominant or subordinate. And the outcome is actually that being subordinate or acquiring a low social rank actually shortens your lifespan. It's not just changing your physiology, will shorten your lifespan, both the median and maximal lifespan. And remarkable for this audience, although it's let's say early stage, I would say it, they also develop early stage atherosclerosis. Now, these are wild type mice. These are not ApoE, these are not LDL knockout. These are not on a heterogenic diet. Yet, at 17 months of age, subordinate animals develop something that can be defined, I think, as early stage atherosclerosis with immune cell infiltration at the commissure and the root of the aortic arch, with some rupture, with some calcification. So this physiological condition is so powerful to elicit an early stage disease, which normally doesn't develop in mice. And this, we are obviously following up on this using proper genetic models. So in conclusion, I know I present a lot of data, but I hope that the key point of this presentation is really that we should look at stress in a very complex physiological and molecular manner. It's a major risk factor for cardiometabolic diseases. The good, if you want, although this is really not a perfectly good, but nevertheless, that this sympathetic activation under certain conditions can protect from obesity. The bad is that at subthermal neutrality, which is for the most part the condition that we study in rodents, it's actually inducing metabolic disorder, increased food intake, obesity, etc. And the very bad is that this is a major risk factor for cardiovascular function, that both elicited by genetic predisposition, chemotherapeutic drug, or natural aging per se. And so I think it's something definitely worth studying, although it's complex and difficult. Number of people collaborated at this project. I don't want to take too much time to go through a large list, but Maria, Carrie, Rachel, Maddie, Cheryl, Valentina, and many others worked at this project in the lab. A number of collaborators, great collaborators. You can do modern great science only by working with the best, and I was lucky enough to convince several people to work with me at this exciting project now and in the future. And I'm not going through all the list. If somebody's interested in more of the social determinants of health, we recently got a research network funded, so please join us and send me an email. I will be happy to link you to all these exciting science we are doing. And of course, this is the funding support that really allowed all this work to be performed, as well as the people that did the project. So thank you very much. I know I've been a little over time, so I'll be okay if nobody has questions and wants to go for lunch.
Well, thank you, Alessandro. This was a fascinating talk. It made me think I really need to reduce my stress levels further. I have a question for you.
Sure.
So, the atherosclerosis studies are definitely ongoing.
Yeah.
But do you expect to find the stress effect on brown fat and brown fat metabolism be part of the phenotype you're going to see?
I mean, it's possible. It's definitely possible. The thing is that we haven't done yet, and we should probably think about an experiment. But again, these are complex experiments. The way to test that, maybe I can unshare my screen. So, the way to test that would really be to test LDL, and this is the strain we're using right now, LDL receptor knockout in thermal neutrality, and see if we can, if the stress-induced brown adipose tissue activation would be able to buffer some of the negative effects. This is possible, but again, it adds another layer of complexity. Something that we are doing right now, and much of our interest has to do with the link between stress, atherosclerosis, and cellular senescence, and how we can manipulate the senescent cells in this phenotype. And so eventually, potentially, that's an option. The prediction being that in the context of stress-induced activation of the brown adipose tissue, you can limit some of the negative consequences, of course, of the dyslipidemia and the hyperphagia and whatever else that you can see at room temperature. So it's definitely something that we can do. We haven't done it yet. Mm-hmm. Hopefully in the near future.
Well, looking forward to hearing about that. So I forgot to say, anybody else wants to ask questions, please raise your hands as usual. I know several people are working on stress now. Okay. It was a really great talk, Alessandra. I think you left no questions to anybody. So
no worries. Thank you very much.
And in the background. So thank you so much for joining us. And
thanks a lot for inviting me.
yeah, so we will be together next week, Friday, as usual. We're going on till the end of the year. And soon we will have a trainee session, just dropping it here, but we'll talk about it when it's really ready. Okay, so have a great weekend.
Bye, everyone.
Bye, everyone.